Welcome, everyone. I'm Allie Wells. I am the coordinator for the Vermont Ag Water Quality Partnership. Thank you so much for joining us today for our Lunch and Learn webinar. Um, we're excited to share that we have a new strategic plan that's out. It's on our website, vtagcleanwater.org. And we have some updated vision, purpose, mission, and goals that I wanted to share with folks today. So for the Vermont Ag Water Quality Partnership, our vision is that there will be a future where Vermont's vibrant working lands serve as a cornerstone of environmental stewardship and conservation, that we're enhancing water quality and wildlife habitat, and these agricultural lands and forestry enterprises will support a thriving economy, foster connected and resilient ecosystems, and contribute to the overall well-being of Vermont and its residents. And our purpose and mission for the Vermont Ag Water Quality Partnership is that we coordinate partner efforts to improve ag water quality in Vermont by providing education, technical and financial assistance to land stewards. And our goals are that we improve water quality, soil health, and wildlife habitat on all working lands, and that we're providing technical and financial assistance efficiently and effectively, and we're delivering high quality service to working land stewards. So if you'd like to read more about our newly released strategic plan, you can find that on our website. And for those who might need a refresher, the partners in the partnership include our federal partners, USDA, NRCS and Farm Service Agency, as well as U.S. Fish and Wildlife Service, UVM Extension, the Lake Champlain Basin Program, our state partners, Vermont Department of Environmental Conservation, the Agency of Ag, Food and Markets, and our Vermont Association of Conservation Districts, and the Natural Resource Conservation Districts, as well as the Vermont Housing and Conservation Board. So with that, I'm going to stop sharing my screen and thank you so much, Eric. Dr. Eric von Wettberg is here from UVM Extension. He's the professor of agriculture, landscape, environment, and is also the chair of the animal and veterinary sciences at UVM, and is going to be sharing with us about managing pasture for healthy farms and healthy soils. So thank you.
Allie, thank you. Everyone, it's an enormous pleasure to be and sincere pleasure to be with all of you. I ran into Allie. It's been a hot minute. We did some amazing botanizing early in the pandemic together, and I was just reminiscing over that. Uh, when we ran into each other at Farm to Plate, it I very much asked for this because part of what I hope to accomplish in talking with all of you today is both tell you about a project we've had ongoing, where we're still analyzing data from some on-farm trials that we've done over the past few years. But more importantly, I hope it's a step towards deeper collaboration. We've loved working with these farmers. Early in this project, we had conversations with some on in conservation districts about sampling together. We ended up not being able to pull it off given some of the samples we were taking, but these are partnerships I really want to steward. Um, so let me briefly outline what I'm going to talk about today. I first want to thank the people who did the work that I'll be presenting, and give appreciation. I wanted to start by appreciating all of you, and um, I'm trying to make this short enough that there's time for active questions. I'm going to then give a brief background on soil testing. The funds from the NRCS Conservation Innovation Grants program were funded by the federal portion of it, have had a strong component of soil testing, and I may have mistargeted the level at which I'm speaking, but I want to talk about some of both the need for basic soil testing services that we've offered at UVM, um, in Jeffords Hall on the main campus, through Extension in the Ag and Environment Testing Lab for a long time. But there's been voices in Vermont who have said these tests are not helpful. They remain central to understanding soil fertility. So I want to talk about both those as well as new tests, some of the places where they have value. And I will try to also talk, at least in passing, about their costs because soil tests do cost money, and there's inherently a trade-off between digging the deepest pit in the world to try to recreate everything about soil versus testing in ways that allow us to make management decisions over larger areas. And I'll end by grounding this in an ongoing project. So um, Ali and Philo Ridge Farm are two of the groups I want to thank. Let me do this more specifically with this slide. The project I'm going to primarily talk about is a project led out of the Vermont Land Trust. The coordinator for it is Annalise Carrington. Annalise is amazing. We've had partners in Extension, particularly Juan Alves. Travis Reynolds at the University of Vermont. He's an economist as well as a maple and beef farmer in the Northeast Kingdom where he grew up. And first a postdoc, Mario Machado, who's now on the faculty at Dartmouth, have been key parts of this. And then we have partners at what was first Biological Capital and closely associated with Philo Ridge Farm in Charlotte. A lot of this work was started by Dr. Lily Hancock. And Maddie Hassert and Noor El Nabusi have both done a lot of the work and planning here. And I'll try to call out their amazing contributions as we go through this. I want to thank a broader team that involves several of our students here at UVM, Alex Steiner, Sadie Southall, as well as a few others who are key parts of this. And I'll try to have my thanks throughout. I'm also going to briefly mention an aligned project that we are in the middle of that we launched off of this. This is a project from UVM's Food Systems Research Institute, led by Professor Gillian Galford, who's a remote sensing GIS and biodiversity expert, as well as a number of others. I think many of these names will be familiar to you. From Ryan Patch to Alyssa White and others, certainly Heather Darby. So it's been a large project looking at some of the farms that are in the first project I'll talk about, as well as conventional farms to understand soil health differences between rotationally grazed and conventional farmland. So with that, I'm going to jump into talking a little bit about soil testing. Now, bear with me. This is, I spend at least some of my time talking to undergraduates, but I think this is an important starting point for how we think about soil testing and why soil fertility testing remains not a waste of time, but something that we need to do and something where, despite those loud voices who say, I have something better, this remains useful to managing our land base, whether it's our most actively used fields or those back 40 pastures that haven't had any fertility applied to them in a half century. If we take a plant, grind it up, and measure all of the different elements in it, plants as well as the animals, whether it's a cow or us that eat them, are dominated by hydrogen, carbon, and oxygen. Life generally takes these up in water or through photosynthesis, and the levels of these in a plant here in Vermont, where we may have drought stress, but it's relatively infrequent and it's not as it is elsewhere. Usually these can limit the growth of a plant, and we need to add water when it doesn't. But it's the other elements that plants need to get from the soil. In order, this is nitrogen, potassium, calcium, magnesium, phosphorus, and so on. Below this list, there may be trace amounts of cobalt or other elements in plants. Plants need all of these, but I've put this up because the relative numbers do matter. There's more nitrogen than anything else that plants take up from soil. There may be small amounts of nitrogen in some of the rock that breaks down based on precipitation and the action of roots to make soils. But most of the nitrogen in terrestrial systems comes from either lightning strikes or from the action of legumes or other plants that associate with nitrogen-fixing bacteria. This means that in most ecosystems, natural or agricultural, adding nitrogen in the form of either chemical fertilizer, legume-rich rotations or manure or other sources is essential to restoring it. And the same goes down this list, but nitrogen tends to be at the top of this list in limiting plant growth. And the way we have thought about this for 150 years is based on chemist Liebig's law of the minimum. We frequently think of this as a barrel that has staves in it of different sizes, and whichever stave is lowest is the thing that most limits a plant's growth. So that might be water or light, but frequently it's something like nitrogen, phosphorus, or potassium. And those three are the most frequently limiting nutrients that plants take up from the soil. And adding them is frequently necessary. They can be added in different ways. It could come in the form of biochar, or it could come in the form of a chemically derived nitrogen-rich fertilizer, but they're frequently limiting. This doesn't mean that other elements aren't sometimes limiting. We are increasingly finding that sulfur is limiting in a range of soils because for much of a century, the burning of fossil fuels in relatively uncontained coal power plants led to the release of sulfur-rich emissions that came back down as acid rain and fertilized our soils with sulfur. As we have cleaned up or reduced the amount of coal that we burn, we have less inadvertent sulfur fertilization, and it increasingly is a limiting nutrient. There are others like molybdenum where plants need a tiny amount. Nitrogen fixing plants require a little bit more, but in general, if a seed has been grown in a place where there's sufficient molybdenum, such as seeds that are usually purchased from production facilities in Oregon or Washington, even high mowing. Well, many companies keep it as a proprietary secret, but a lot of our commercially grown seeds are produced in places like Oregon, Washington, or Chile and South Africa. If those soils have sufficient molybdenum, the seed itself has enough molybdenum for the plant to be grown without any molybdenum deficiency. To study molybdenum deficiency, you actually need to grow plants for a generation or two in the absence of it, and then study the molybdenum deficiency because usually there's a sufficient amount in the seed for it. And it's very expensive to over fertilize with molybdenum because it's so rare in the Earth's crust. But this framework is important, and it's something that I think went into the mind of the RFP writers at NRCS for conservation innovation grants. The kinds of fertility testing that the Ag and Environment Testing Lab, the extension managed soil testing lab provides is essential. If we take biomass, that could be a harvest of corn, it could be our hay, it could be the milk in a dairy herd in a grass-based operation. If we take biomass off of a field, it will need to be replaced the next year. If we don't replace it, the yield will go down. And eventually, if we leave fields fallow, a little bit of it may come back. Lightning strikes may bring back a little bit of the nitrogen. The breakdown of the rock underneath may restore some of the other micronutrients, but we can maintain yields with sensible fertilizer application by tracking this. Increasingly, we do offer a broader range of tests. The Ag and Environment Testing Lab has offered micronutrient tests to look for, so that a farmer or a manager can help look for micronutrient deficiencies. Micronutrient deficiencies are real things, but they're far less common than nitrogen, potassium, or phosphorus deficiencies. There are other attributes of soil that do very much matter, and I'm going to talk about some of these, but soil physical structure, particularly compaction, can make it harder for roots to get into the soil and limit their uptake of nutrients that may be there. We're particularly interested in soil aggregate stability because it's a good integrated measure of many of these. The biology of a soil, particularly the microbes that are present in it, the bacteria, maybe more importantly, the fungi that are present, are also essential because in many cases, plants can directly take up nutrients, but they often do it more efficiently by giving photosynthate, the sugar from photosynthesis, to mycorrhizal bacteria, mycorrhizal fungi, pardon me, a rhizobial bacteria, or other microorganisms that are more efficient at taking up those micronutrients. And we do have a range of emerging testing options for those that can allow one to sensibly increase the value of these. All right, so the most widespread package for this was developed at between NRCS and Cornell. This is called the Comprehensive Assessment of Soil Health Package. Since this is being recorded, this is easily found. I suspect many of you are as familiar or more than I am with this. But this gives a complete snapshot of soil health. It has some of these integrated relatively new measurements that I mentioned. It includes many of the attributes of a standard soil test, your pH, your organic matter, your micronutrients. It has the aggregate stability of the soil, how well it holds together. It will measure something called active carbon, which is technically the permanganate oxidizable carbon, but it tells us not just how much organic matter is in the soil, but how much of the carbon is adsorbed or attached to the clay or sand particles in a soil, which is a better estimate of how well that carbon will remain in the soil if it's plowed or due to microbial respiration. And it can also, if you use a penetrometer, give you a rough but imperfect estimate of how hard the soil is. At UVM, in a new lab called the Soil Health Research and Extension Center, that's adjacent to our Ag and Environment Testing Lab, we have the capacity to also implement many of these tests so that we can keep these resources here in the state and provide options to farmers who may not want the whole package and provide advice that may be a little bit more tailored to our uniquely clay-heavy soils. Right. So with that brief interlude on the value of the testing services that I'm not in either of our testing labs, but work with them closely on projects like this one that I'll jump into now. So I'm going to shift from just talking about soil to talking about grazing. I grew up in upstate New York. My apologies for that, but I can't hide that in my accent. I grew up in a place called Hamilton. I grew up behind a dairy farm. I used to walk through their back pastures as a kid when I was hiding from Sunday school. I admit to that. I love to see animals grazing, and I've seen over my middle-aged life plenty of small family dairy farms transition to other uses or consolidate. We work from the premise that grazing operations, if they're well managed, can lower the cost of inputs, the cost of going into business in ways that both are economically viable, ideally lead to socially sustainable and viable livelihoods, but also increase soil fertility, protect water quality, sequester carbon, encourage biodiversity, and have benefits to the farmers themselves while providing nutritious food. This sounds great, but I am a scientist and I think we should be skeptical about claims that grazing is a magical process. So we set out with our partners at VLT and Philo Ridge to investigate this with the support of NRCS. In this project, we've evaluated ecological, social, and economic outcomes. Today, I'm going to keep some of the results short because we're in the process of doing analysis still, and my partners, Maddie and Noor, are going to present in more depth at a conference next week, and I don't want to take away the thunder of our amazing staff. When we went into this work, based on a literature review, we know that there's been interest in grazing in the Northeast for decades, but there's been a lack of recent robust research with proper controls that has investigated it. And integrated research is hard to do. Lots of ideas. It's wonderful to dig a soil pit. You definitely learn a lot. It's incredibly costly to do so, and frequently the benefit of what you learn doesn't outweigh the cost. A lot, this can be said of a lot of practices around farm management, and we wanted to know what it costs to be able to give the kind of information to the Vermont Land Trust so that as they work with conserved land under easement, they understand what both the environmental consequences of land going to grazing and grass-fed animal production is, but so that they can also help young farmers enthusiastic about stewarding the land to navigate the challenges of raising capital to transition towards a grazing operation. Furthermore, we wanted to ask why our farmers are farming. What do they love about it? What keeps them in it when it is not the easiest thing to do? So we've worked with at least 15 farms across Vermont, as well as some control farms. We've run this project over 4 years, and the farms with which we worked, I'm gonna be circumspect about giving their names since this is gonna live online, but they're a mix of beef, dairy, and sheep farms and some integrated operations. And the number of fields we've had has actually gone up. We took a number of fields in the first year when we had approximately 100 and have subdivided them because as we've worked more closely with our farmers, found that they might be managing one field in multiple ways. And for the analyses we're doing, they've been best broken up. In this project, we used NRCS practices, and we were able to pay over half of this award has gone directly to our farmers as direct payments. About 65% of it has gone directly into the pockets of Vermont farmers. In this, they have received payments for supplemental seeding, for nutritional amendments. This might be an organic fertilizer, it might be wood ash, it might be biochar. The core process that all of them have done is in this project, each of them took new fields under management. They might be leased fields next door or fields they were already in the process of buying. And they have transitioned them to managed intensive rotational grazing. By merg, I mean a process where the cows or sheep are moved every 12 to 72 hours to fresh grass so that they are given time to eat not just the tastiest vegetation, the cup plant or the tastiest of the grass, but they're left on it long enough that they take down all the vegetation so that the field doesn't transition to less palatable, weedier species, and so that the animals are getting balanced nutrition from all of those herbaceous plants. In our initial proposal, we had intended to study deep tillage. This is a practice that has received a lot of attention. We worked with NRCS and they advised us that we would not be able to get approval for it. And most of the farms we were working with, a few have done it anyways on their own initiative. I have done it on one of our university fields, and I have come to believe that rip sowing is something that no one should do in the vast majority of cases, that in most cases, the tire pressure actually causes more compaction. The cost of the annual mix may be worthwhile in the short term, but usually leads to drawbacks. And based on the research of Joshua Faulkner in a paper he published with Juan Alves and others last year, that sorghum Sudan grass accomplishes as much compaction remediation without the drawbacks of heavy fossil fuel use or sort of snake oil biostimulants, and that the alleviation it produces as a bio drill is more uniform across the depth of soil. And this is based on Joshua's peer-reviewed um, research with multiple controls over 2 farms on different soil types. All right, so this is I have delved into a handful of things. My I've said this earlier, but I want to reiterate this. My hope is interesting conversation and what I hope is productive future collaboration. All right, so I'll jump into a few of the things that we've done. We sampled pasture species diversity 3 times through each of the pass-through growing season to collect botanical data. We also took forage quality samples that were analyzed at the Bio 1 lab. We Dairy 1 lab. And then in the first year and the last year, we took soil samples to estimate soil physical and chemical characteristics. Because each farm had different practices in each field, each of the farms worked with a technical service provider to improve their rotational practices and to use the first year's data to guide fertility additions and seeding practices in future years. That means that each field in this has received a different experimental treatment and had a different starting point. So we have a before and after experimental design with each of the approximately 100 fields having a different treatment. So we're in the middle of using an approach called structural equation modeling to sort through this to understand the relationship of different variables to one another. I won't be showing results from this. Alex, the doctoral student working on this, is making great headway on this. We used the cache results from the first year to assess how the soils in the fields in this study compared to soils across the region. In a finding that I think will surprise few of you, the hardness of a lot of our pastures was quite high. This is consistent with a lot of grass-fed operations being on land that is less suitable for row agriculture, for producing silage corn. So these are harder fields, often a high clay content, and that's not surprising. Many of these were hay fields in the past, and as a consequence of that, they have high levels of carbon. So these active carbon measurements are pretty high, and they are generally otherwise pretty fertile. They're not deficient in minor elements, and they're not extreme pH outliers. So I've highlighted the soil hardness. They're generally respiration of soils, how much carbon dioxide they emit under lab conditions, is a good proxy for how much microbial diversity they have. So these are generally biologically rich soils. The active carbon and soil protein also both support that conclusion. We didn't see huge differences in the amount of carbon between our negative control fields. These are fields that were in hay or silage corn that have remained in that throughout the study. And we have a set of positive controls. These were fields that were already in managed intensive rotational grazing before the study. The fields we brought into this study are more variable in how much carbon they have and in other characteristics, but they're not substantially or significantly different in that. One of the areas where we hope to roll out useful results soon is through this study, we realized we needed to measure the amount of the yield of a field, how much biomass is being produced. Our colleagues at Organic Valley and Planet Labs had already been developing some methods from this, so we began working with them and have continued this to use remote sensing to estimate pasture biomass. For those of you who have gone out and clipped vegetation, it is a time-consuming activity, and it would be impossible in a project of this scale to do that over 100 fields on 20 farms before each of them is grazed. It's not logistically possible. And as a management tool for farmers, tools like rising plate meters or grazing sticks can be helpful guides. But if this can be automated from remote sense data, it could save farmers time and in many cases be a more accurate way to detect fields that should be grazed, fields that should be left to produce hay, and those that need to rest and regenerate. Tied to this project, we've been taking estimates of biological diversity in our field. So this graph comes from the related Food Systems Research Center project with Bailey Kretzler and Gillian Galford. With our soil samples, we took a subset of them, we extracted the DNA from them, and we used this to estimate the bacterial diversity in the samples. The set of farms here is a slightly different set of farms. This is 3 conventional farms in Vermont and 3 grazed farms from our study. You'll see that the amount of diversity is roughly equivalent. It varies through the season, and it actually tends to be a little bit higher on conventional farms than grazed farms. This is one indication of how diverse they are. This on its own doesn't tell us about the function of that diversity, however. And this is part of where a lot of new tests of soil biological activity have a lot of power, but are easily misinterpreted. What we think is in some ways the best indication is not how many bacterial or fungal or nematode species are there, but what's the length of the food web? How many different ecological functions are performed by the species? So in a paired data set, we've also collected nematodes from each of these fields and characterized them. Nematodes have a range of ecological functions. Some are parasites of plants, nasty little things. Some of them eat fungi or bacteria in the soil and actually perform an important ecological service for us. Others can be animal parasites. The diversity of nematodes is a very good indication of food web length. And within this, over 2 different times of year, we see substantial differentiation of grazed and conventional fields. We're in the middle of analyzing this. I have the paper open and I'm still scratching my head at some of the results. I am hopeful that we'll have a clear story that emphasizes this point that bacterial diversity often is associated with nematode diversity. I realize I'm not showing that here. That there are not overall diversity number differences, but there are community structure differences between conventional and grazed settings, and that these can give us insights into some of the ecological functioning that differs between a conventionally managed no-till cover cropped silage cornfield and a grazed field, even if the two may have equivalent numbers of microbes and similar amounts of soil carbon. All right, so across both of these projects, a key part of what we've been doing is in trying to improve the means, the sensitivity, the thoughtfulness by which we report back to the farmers with whom we work, the data that we've been able to collect with their help over the last 4 years. So an example of this that I'll give is some of the plant species diversity that we have. As we think about this, I feel busy every day, but anyone who's milking several times a day is inherently busy and has only so much bandwidth to interpret things. So we've been trying to report out the the botanical diversity, breaking out communities into grass, legumes, and forbs and other forbs. All of these are useful for the diets of livestock. Um, we're further breaking this out. We're still working on how to present this effectively in ways that break out how palatable the grass species are, and in some cases the forbs are, so that we can help farmers understand how seeding choices or choices about how big their paddocks might be or how long they leave their animals on a particular paddock determine that. One of the students with whom I'm working, Helen Goodyear, is digging into this in a subset of farms to really try to answer the question of how high should the stubble be in a grazing setting to maximize plant species diversity and yield. All right, so we've tried to fully integrate not just taking data on what farmers do, but using surveys, interviews, and stakeholder events as well as farmer panels to understand farmer perceptions, what drives the decisions they make, and to compare what we observe when we collect data from them and their own perceptions about how fertile their fields are or how they would make decisions based on the information we provide them. Some of the takeaways from this, as with everything in this project, this is work underway. The farmers we work with are amazing. It is humbling and a complete honor to work with them every day. They're very intentional about choosing to farm, the decisions they make, the love they have for the land that they steward and walk on. And it is interesting how many of them, particularly those who are smaller, those who have less or no hired labor, they underestimate their own time. And to be fair, I have some sympathy for this. It's not unusual for me to be up at 2 o'clock in the morning trying to work on a student's manuscript so that it can go into peer review so our students can be published. One of our hopes from this, this is work underway, and I admit I'm not entirely sure how to advocate for it, but in many cases, NRCS payments or other payments for ecosystem services need to be higher because our farmers are underestimating their own time, and their time is valuable. All right. So we've also been taking information from our farmers on monthly records of how much time they spend doing different things, the cost of infrastructure. We've been using an NRCS approach. This is one of our farmers. I apologize for exposing Guy here, but it's well known that Guy has worked with UVM for over 20 years and with the Vermont Land Trust. So I've used his intentionally, but I did mean to black this out. This is a T chart that shows the cost of implementing something as well as its benefits. One of the things that this farmer has particularly found, but this holds up across our project, is the steps we've worked with to implement practices that it can increase yield on a number of the fields he took under management in this project have allowed him to lower the cost of additional feed, and this has substantial benefits. This is a particularly interesting farm because their milking regime is a little bit different than elsewhere. This is one of the farms that was already in the process of rip sowing, and this is a place where some benefits emerge. It's a particularly flat field, and the underlying geology here is less problematic. I still suspect that the overall compaction has actually not decreased. But this is how we're looking through costs of production. Some of our takeaways here are that saved feed costs are substantial. And this supports the premise that I know John Winston, among many others, have had for well over a decade, if not several decades, that moving towards rotational grazing can be a way to lower costs of production in ways that when commodity prices for milk or for meat are at points that are disadvantageous to smaller producers, there's a potential to still be economically viable. With this, and I say this as someone who works with economists but is not one, the devil's in the details. Factors such as the cost of accessing land, costs of financing, and costs of payments for services or NRCS payments really do matter when margins are tight. And something can go from being ecologically beneficial and economically beneficial to not with small changes. All right. So I've been thrilled to have the opportunity to present this, even if we're not done and still we're doing our best to be good stewards of the federal support we've received and the trust that we hope we have earned from the farmers with whom we've worked. Some of the other outputs, besides talking in places like here, is we've done a number of pasture walks and look forward to doing more. We'll certainly be reporting to NRCS. We've started putting out some case studies and with the permission of our farmers and stakeholders, hope to put out more. And there's a handful of journal articles that we have put out and many more in process. So I think I've stayed within what I hope is reasonable time and left time for discussion. I'd like to thank all my partners again, particularly with these slides. I've had a lot of support from Maddie, um, Mario, Noor, Alex, Travis, Sadie, um, our technical service providers, Juan, Cheryl, and Tom have been fantastic. Thank you to the support from Philo Ridge, NRCS, and thank you for your attention. Provide thanks to a few of the students who were with us at the start, and I am happy to use time to answer questions or talk creatively and even brainstorm ways to work together in the future. Allie, thank you again.
Thank you so much, Eric. That was great. Folks, if you'd like to raise your hand or put any questions in the chat, we can address them. And Ben Gabos, I see you put a link in the chat. Did you want to mention anything about that?
This is the
No.
paper I mentioned.
Yeah, right.
Thank
Yep.
you, Ben.
Yep.
And I'll put into the chat a few of the others that have started to emerge from this while we wait for questions.
I guess I have a question that comes to mind with different tools around soil sampling available. Do you foresee any need to change the recommended soil types of tests that are used in developing nutrient management plans in the future so we have more tailored plans?
I'm going to defer that question in the future to Joshua Faulkner, who I trust, and Heather Darby, who I respect and trust immensely on that. The short answer is I think we're doing pretty well on nutrient management planning. And I'll admit there's aspects of that where I don't, where I think many of you who are doing it have as good a sense as I do. I think part of where I'd go with it is, and this is something we actually wanted to do in this project. I'll get to your, I'm going to go around the question for a second. One of the things we wanted to know, and I think we do need to know in Vermont is, um, phosphorus is one of the most critical. Over the past 2 decades, occasionally the term peak phosphorus has showed up in the literature in a range of soils, and some ours are prone to this. Phosphorus can be as limiting or more limiting than nitrogen. Most of the global sources we use for phosphorus in fertilizer they come from Florida, they come from Morocco, Algeria, Tunisia. There are not that many natural rock deposits that are rich in phosphorus, and many of those deposits, particularly in North Africa, happen to be laced with heavy metals. And the phosphorus is good, the heavy metals are bad. That means that keeping the phosphorus we have in systems is important. I'm saying this to people who know this as well or better than I do, but the nature of phosphorus in soils is it tends to end up attached to soil particles. Some plants are good at getting those off of soil particles by basically mining it off in cluster roots, but by and large, mycorrhizal fungi are more effective than the plants are at getting at it. They have the hyphae are smaller than roots. The fungus, its biochemistry for getting it off of particles is simply better. There's an argument they've been doing it longer than plants have, and we only have plants on land because of mycorrhizal fungi. That said, mycorrhizal fungi don't like tillage. And what one of these tests the PFLA test in particular. It's a test that measures fungal to bacterial ratio based on their phospholipid profile. What it basically tells us, how much has this field been tilled? Most mycorrhizal fungi just won't grow well in a tilled soil. When we use no-till and cover cropping, we can encourage them in an annually cropped system. It comes with the drawback that most no-till is going to require the use of an herbicide to terminate the cover crop so that you get reasonable establishment of the cover crop. But it can still be mycorrhizal friendly and phosphorus friendly. Those tests, so new tests like PFLA are useful for telling you whether the field's tilled or not. In terms of developing the management plan, the richness of that biology and the extent to which phosphorus may be available symbiotically from mycorrhizal fungi, rather than needing to come more directly from the soil or fertility you add to it, can change management decisions. But in terms of testing for the overall amount there, I think the way we test is perfectly fine, but I would admit that I defer to others. I think, well, this is again citing Joshua's work. I have found Joshua's work on tile drainage fascinating because in hindsight, it makes sense, but tile drainage does amazing things for extending season length. For getting onto fields that are too wet, it's can be incredibly helpful. From a permitting perspective, when you're putting channels in the soil that are going to send phosphorus out of the fields and downstream, it's something that we should be thoughtful about. But in deference to others, my long answer is I think we're testing for the right things right now. But feel free to contradict me.
Thank you. Any other questions? Also, feel free to just unmute. Well, I was also going to ask about a couple other things, but it sounds like with the presentation next week that there'll be a lot more information coming out. So could you mention a little bit about where that's happening?
That is happening in Manchester, New Hampshire at the Northeast Grazing Conference. And I hope we'll be through a few more things. We have the size project where, to be fair, Alex will be working for the next 2 years on this as part of his dissertation. And one of the first thing I learned is I actually never finished my master's degree. I did the work for one and then went straight to a PhD without actually submitting the document, which I regret regularly, but neither here nor there. I have the job I always wanted. Um, it always takes researchers as much time from the point they've collected the data to make sense of it and get it through peer review. We're we realize we've been supported by NRCS. I'm happy to talk about the drawbacks of the academic publishing model we have. We have tried to be very thoughtful about putting information in the hands of our farmers as quickly but as thoughtfully as we can, and that doesn't require peer review. But peer review is kind of like democracy. It's a messy process with biases in it, but it also really works to validate experiments. It gives a chance for things that are good ideas to emerge, to fight against being called snake oil when they might be called that, but also to take things that you simply can't validate, and expose them. So we do want to put this work through peer review eventually. It's just we're taking some time, and the structural equation modeling won't be done in a week. Maddie and Nora are going to present a few more results on the pasture biomass, perhaps the forage nutritional value, and a few of the soil properties. Um, I hope within the next year there's much more, and um, we're committed to data openness, so while protecting what we can of farmer privacy.
Thank you. I also, for folks, put a really brief survey in the chat, which helps us to follow up with any additional questions you have and put together more lunch and learns that may be of interest. And Eric, did you put your contact information in
I will put it in
there?
the chat. I did
In
not
that.
have it on my
Okay.
slide, but I certainly should. I have a distinct surname. If you Google von Wettberg, I am easily found, and we work for the public at a public university, so I'm easily contacted there.
Thank you so much.
Everyone, thank you for your attention, and feel free to reach out with thoughts, concerns, future projects, and enjoy the cold weather. Allie, thank you.